we're going to start here with our events of vision. And again, you're going to want that special senses handout that was available to you in Blackboard. So the first thing we have to do in order to be able to see something is for our eye to move toward that object. Okay, I could have the best eyes in the world, but if they don't have the ability to move and look at, quote unquote, move towards whatever object I want to see, I'm not going to be able to see it. And that's why, keep in mind, an eyeball is like an inch and a half in diameter. It has 6 muscles connected to it that allows for very, very precise movement. Okay, we have our 4 rectus muscles, which are straight. We have our 2 obliques, which are at an angle, and that allows your eye to move very precisely to look at whatever object that it is that you want to see. We are able to see objects because light reflects from them. The light that reflects from them then is sent towards our eye. Okay, and these yellow lines that we have to the left of this diagram is representing the light reflecting from an object. The first thing that part of the eye that light is going to come in contact with is the cornea. And the job of the cornea is to refract light. Refract is a fancy word for bend. And if you follow those arrows from left to right, you'll see that a lot of these structures that we're going to talk about, their job is to refract light, to bend light, because we want to focus and concentrate that light so it hits in that central fovea of the macula so that we're going to have the best possibility for the clearest vision. So the light is going to reflect off an object, it's going to pass through the cornea and get refracted. It's then going to pass through what's called the aqueous humor. The aqueous humor is, and if you look at the word aqueous, watery, a watery substance that can be found from the iris, between the cornea and the iris. Okay, and its job, again, is to refract or bend light. I shift to this diagram. Again, it's got that same kind of thing where we're bending, refracting the light, because it's now going to show you the pupil, right? Because, again, that's another reason we want to refract that light through the cornea and the aqueous humor so we can concentrate that light so it's going to be able to pass through the pupil so that we'll be able to use that light to process information. So the pupil is going to regulate how much light is going to enter the eye because, as we talked about, it can get larger or smaller depending on light conditions. The brighter the light, the smaller the pupil. The dimmer the light, the larger the pupil. We are then going to have aqueous humor between the iris and the lens. So we're going to have the aqueous humor in front of the iris. We're going to have the aqueous humor behind the iris, but in front of the lens. And again, its job is to refract the light. Next, Next we have the lens. Again, it's there to refract and focus light. And it is controlled by the ciliary body, which is that pink kind of fluffy looking stuff just above the lens and just below the lens. And it is kept in place and controls the lens, the shape of the lens, through those suspensory ligaments. If the suspensory ligaments pull on the lens, it flattens the lens and causes the light to be bent one way. If the suspensory ligaments and the ciliary body loosen up on the lens, it bulges, which causes the light to refract in a different way, depending on whether you're looking at something close up or in the distance. Once the light, sorry about that, I keep thinking I'm going to go to the next slide. Once the light has passed through the lens, the posterior portion of the eye, kind of looks at an orangey color there, is filled with something called the vitreous humor. The vitreous humor is similar to the aqueous humor, but it's more jelly-like. It's got more body to it. And again, that helps to keep the shape of the eye. However, it's also going to, again, I'm sick of saying it, you're sick of hearing it, it's going to refract that light, so we're focusing that light, that we're concentrating that light, so it's going to hit the photoreceptor layer exactly where we want it to hit. Okay, now this is a nice diagram because it's showing us 1, 2, 3. That's our layers of our neurons that make up the retina. Number 1 is our photoreceptor cells. Number 2 is our bipolar cells. Number 3 are our ganglion cells. And what's weird is you got to follow the arrows. The light is going to come into the eye and pass through and not stimulate the second or third layer because they're not photoreceptor cells. The only cells that are going to be affected by the light are that first layer, the rods and the cones. So the light's going to come all the way to the back of the eye. It's going to stimulate those rods and cones, initiate a potential change, that action potential that we talked about, and then the signal is going to be sent in the opposite direction from the rods and cones, our photoreceptor cells, to our bipolar cells, to the ganglion cells. So we're going to transmit that impulse from one layer of cells to the next. And this is another similar diagram, okay? The light's going to go all the way through, stimulate our photoreceptor cells, and then the action potential is going to work its way back out. Now, the axon of those green cells, of the ganglion cells, will be bunched together and form the optic nerve. And that's the nerve that's going to carry that information, ideally, ultimately, back to our occipital lobe. Now, because we have contralateral control, opposite side control, many of those fibers from the right eye are going to go to the left side of the brain. Many of those fibers from the opposite eye is going to go to the opposite side of the brain. That's what we call the optic chiasm, that crisscrossing, right? We talked about decussation in the spinal cord. This is sort of kind of like that. We're having our nerves crisscross from the right eye to the left side, from the left eye to the right side. We then follow our tracks back, and ultimately we land with our information being funneled back to the occipital lobe, specifically the primary visual cortex of the occipital lobe that's going to interpret the visual signal. It's going to allow you to know what in fact you are looking at, what you're seeing. Okay, now that's vision. Next we have hearing. I think the ear is super cool, all right? It's very linear. One structure affects the next structure, which affects the next structure, which affects the next structure, okay? Again, we have our information in that little packet that I had you print. The pinna is the ear, the flappy part that we would refer to our ear. It's not terribly functional for hearing, but it does act as a kind of a funnel to take all of the sound and bring it into our external auditory meatus. Now you'll see that your ear is divided up into 3 regions: the outer ear, the middle ear, and the inner ear. And your handout shows you what structures are located there. From the tympanic membrane out is the outer ear. Right, so we have the pinna, we have the external ear structures, we have your external auditory meatus, and the tympanic membrane. The inner ear is the cochlea and the semicircular canals. The middle ear is what's in between, and then in this diagram, it's that green color. And that's where we're going to find the bones of the ear, the malleus, the incus, and the stapes, and the auditory tube, which used to be called the eustachian tube. That tube is going to allow fluid to drain from your ear down into the back of your throat. Now, when sound, how your ear works is the sound is waves, all right, is movement, right? And that's what we see here, that red kind of squiggly looking guy. Those are sound waves, air movement. If you don't believe that sound is caused by the movement of air, talk and put your hand in front of your mouth and you're going to feel the air hitting your hand, okay? So your external ear, right, your pinna and such, funnel those sound waves down into your tympanic membrane. The tympanic membrane moves, it vibrates. Okay, it's going to move back and forth. Well, this is kind of cool. The malleus is leaning on the inside, leaning up against the tympanic membrane. So if the tympanic membrane moves, what's going to happen to the malleus? And remember, the malleus is leaning against the tympanic membrane. If I move the tympanic membrane, the malleus is going to move. Next in line, we have the incus, which is leaning up against the malleus. So if I move the tympanic membrane, it causes the malleus to move. If I move the malleus, it's going to cause the incus to move. And you guessed it, if I move the incus, that's going to cause the stapes to move. Okay, and the stapes is our third bone. Now you'll see in this graphic that the stapes, quote unquote, sits on top of the oval window. The oval window is a membranous structure. So when the sound comes in and moves the tympanic membrane, which moves the malleus, which moves the incus, which moves the stapes acts like a piston. It's going to be moved in and out of that oval window, which is going to cause the fluid in the cochlea to be shifted back and forth. Right, and again, this is just another not so great diagram, but it's showing you that the stapes is connected to the cochlea, and the cochlea is the part of the ear that's going to allow for hearing. Inside of the cochlea, we have these structures. We have two canals, the vestibular and tympanic canals, which are filled with fluid. We also have the cochlear duct, which is filled with fluid, and the organ of Corti. The organ of Corti is really what's going to allow for hearing to happen. Right, so here we have it. We've got our sound coming in. It's going to move the tympanic membrane, move the malleus, move the incus, move the stapes. The stapes acts as a piston. Our cochlea, where we're going to find our vestibular and tympanic canals, and our cochlear duct filled with fluid, that's contained within the cochlea. So here's our stapes. Here's our oval window. When we move that stapes back and forth, the fluid, the perilymph in the tympanic duct and the vestibular duct will move, which is going to cause the fluid in the cochlear duct, which is called endolymph, for it to move. When that fluid moves, it's going to cause the cells of the organ of Corti to be stimulated. That's going to send a action potential out through the fibers of these sensory neurons that you see. That's the vestibular cochlear nerve, or the auditory nerve. And that very simple process of one structure leaning on another, causing a little bit of movement, allows for you to hear all of the rich sounds that you hear.